Hello friends, photoluminescence is a process whereby certain materials absorb electromagnetic radiations such as ultraviolet, visible or infrared light and gets excited, then release energy in the form of light whose wavelength is longer and energy is lower than the absorbed light principle. When these materials absorb light, their electrons are excited to a high energy state. As these high energy electrons are unstable, they return to the ground state. As these high energy electrons are unstable, they return to the ground state. During this relaxation process, some energy is lost as heat and the remaining is released as light whose wavelength is longer and energy is lower than the light absorbed. The difference between the wavelength at which these materials are excited and the wavelength at which they emit light is known as Stokes shift. Photoluminescence is of two types. Number one, fluorescence. It is a fast process in which light emission occurs rapidly, typically within less than 10 to power minus 6 seconds. Number two, phosphorescence, in which there is delayed emission of light that lasts for seconds or even hours. It is a slow process. Hello and welcome to my channel, Biology class. Friends, today we will discuss about the principle of fluorescence microscope and its applications. Fluorescence was first discovered by British scientist Sir George Stokes in 1852. The first functional fluorescence microscope was developed by German physicists Otto Hempstead and Heinrich Lehmann in early 1990s. This figure illustrates the principle of fluorescence microscope, which operates based on the principle of fluorescence. In fluorescence microscopy, specimens are stained with fluorescent dyes known as fluorophores that binds to specific structures such as proteins or nucleic acid. When these fluorescently labeled specimens are exposed to short wavelength light, such as ultraviolet or blue light, their electrons are excited to a high energy state. The high energy electrons, which are unstable, then return to the ground state, releasing energy as long wavelength light. This is known as fluorescence. The long wavelength light is then captured by the eyepiece and the detector, which forms a high contrast magnified image against a dark background. Fluorescence microscopes often do not have traditional condenser because they function via epiillumination. In this process, the objective itself acts as the condenser, focusing light onto the specimen and then collecting the emitted light. Such type of microscope is known as epifluorescence microscope. Typical components of a fluorescence microscope are a light source, excitation filter, dichroic mirror and the emission filter. The light source, which transmits high intensity bright light, is usually a xenon arc lamp, a mercury vapor lamp or a high power LED. The high intensity light transmitted by the source passes through an excitation filter. This filter allows the passage of only short wavelength light which is required to excite the fluorophore. The short wavelength light then falls on a dichroic mirror from which it is reflected down and passes through the objective and is then focused onto the specimen. The fluorophore with which the specimen is labeled with absorbs the short wavelength light. As a result, the electrons are boosted to a high energy level. When these unstable high energy electrons return to the ground state, light of long wavelength is released, which is then captured by the objective. From the objective, the light of long wavelength then passes through the dichroic mirror and then through an emission filter. The function of the emission filter is to allow the passage of required long wavelength light, which is then captured by the ocular and the detector, which forms a high contrast magnified image of vibrant bright colors such as red, green, blue or yellow against a dark background. In fluorescence microscopy, different fluorescent dyes are used for different purposes. For example, acridine orange is used to stain both DNA and RNA. It is frequently used to identify microorganisms. Another compound is phalloidine, which is used to label actin filaments in mammalian cells. This component is frequently conjugated with fluorophore like Alexa Fluor 488. Another important compound is green fluorescent protein, which is used for real-time tracking of proteins in living cells. In addition, other fluorescent dyes such as Alexa Fluor and rhodamine is used for labeling specific proteins. 4 applications. Number 1. The technique of fluorescence microscopy is extensively used in cell biology and molecular imaging. Number 2. It is also highly useful in medical diagnostic and research. In addition, fluorescence microscopes are widely applicable in material science and mineralogy and also in food quality control. Friends, this is all about the principle and applications of fluorescence microscope. For more updates, subscribe my YouTube channel, Sam Biology Class.